All right, party people, we are back at it with another one. Let's get it. So, here we have a question asking, which of the following precursors most likely contributed to normal glycemia while fasting? Below is the list. Please make your choice. 3, 2, 1. Peekaboo. All right, let's go through why the correct is correct and why the incorrects are incorrect. Now, for reference ranges, normal range for blood glucose is 72 to 99. Impaired fasting glucose, or prediabetes, is 100 to 125. Diabetes is above or equal to 126 on two separate readings. Now, that's basically, and the question is asking, how do you maintain normal glucose levels after 24 hours of fasting? So, first and foremost, during the first 12 to 18 hours, your body is using glycogenolysis to maintain your blood sugar level. The primary source for glucose during this time is glycogen. Glycogenolysis is the breakdown of glycogen into glucose. This pathway typically sustains glucose for about 12 to 18 hours. There's no strict cutoff. Just know that after about a day, glycogen stores are depleted. You will not be using glycogen 3 days into a fast, for example. The key enzyme that you need to know for this is glycogen phosphorylase. This is active during fasting. It's phosphorylated when it's active. And yes, your licensing exams do require you to know this for whatever reason. It might seem strange, but you need to know it, so you're going to know it. Just make a note of that. Now, after glycogen has been depleted, your body flips the switch into gluconeogenesis. Once glycogen is exhausted, the body shifts into gluconeogenesis, making glucose from non-carbohydrate sources. The primary substrates used in this process are lactate, glycerol, alanine, glutamine. All right. Now, in terms of lactate, this is converted into glucose via the Cori cycle. So in muscle, you have glucose, which is then converted to pyruvate, and then pyruvate into lactate. This is via anaerobic glycolysis. In the liver, the lactate is converted into pyruvate via lactate dehydrogenase, and that pyruvate is then converted into glucose via gluconeogenesis. It's important to note that you do not go directly from pyruvate to phosphoenol pyruvate. This is due to the irreversible steps in glycolysis. So instead, what happens is you go from pyruvate to oxaloacetate. This is via pyruvate carboxylase. This requires biotin B7. Now, acetyl-CoA is a positive allosteric regulator of this step. Oxaloacetate is then converted into PEP via PEP carboxykinase. PEP is then eventually converted into glucose via the gluconeogenic pathway. Now, in terms of glycerol, glycerol can be converted into glucose. Now, please note that triglyceride breakdown involves glycerol as well as 3 fatty acids. The fatty acids cannot become glucose. Glycerol, however, can become glucose. So glycerol converts into glycerol 3-phosphate. This is done via glycerol kinase. Glycerol 3-phosphate gets converted to DHAP, and DHAP is then subsequently converted into glucose. The key takeaway here is that glycerol is a gluconeogenic substrate. Now, in terms of amino acids can be converted to glucose. Most amino acids are glucogenic. That means that they can become pyruvate or other TCA intermediates. The exceptions, making them strictly ketogenic, are leucine and lysine. These can be converted to acetyl-CoA only and therefore cannot become glucose. Some amino acids are both ketogenic and glucogenic. However, don't worry about memorizing them because they don't really get asked that much. The key gluconeogenic amino acids that you need to know are alanine and glutamine. All right, now dealing with alanine. Alanine can be converted into pyruvate and subsequently glucose. This is done via the enzyme alanine transaminase, ALT. This requires B6. So the reaction that you need to know is that alanine plus alpha-ketoglutarate gets converted into pyruvate plus glutamate. This is how alanine contributes to gluconeogenesis. Now let's go through the other incorrect answers and break those down. Acetyl-CoA is not gluconeogenic. It is a precursor for ketone bodies such as beta-hydroxybutyrate, acetone, etc. This is formed from ketogenic amino acids such as leucine and lysine, and therefore it is the incorrect answer for this question. As far as valine is glucogenic, but not a major gluconeogenic substrate. Therefore, this is not the best choice for this question because it's testing primary pathways. Essential fatty acids such as linoleic acid, they are required from your diet. They cannot be converted into glucose. And often on your licensing exams, they use this essential fatty acid as a distractor. As far as stearic acid, this is a saturated 18-carbon fatty acid, not gluconeogenic. All right, let's spin the block and recap everything we covered. After 24 hours of fasting, the body switches into gluconeogenesis. The main substrates are lactate, glycerol, alanine, and glutamine. Now you have to know that alanine via ALT gets converted into pyruvate and glucose. All right, that's it for this one. Go crush it.